Okay, 2 o'clock, time to get going. I believe the last thing we discussed was the start of our unit on cancer. Is that correct? We talked about the 2-hit model and genetic load and aging as the number one risk. Okay, so just to reiterate something, cancer is a game of numbers. It's not a guarantee. You can live the most risky lifestyle imaginable and still not get it. You can live the safest lifestyle imaginable and still develop it. However, it's just like any other thing involving chance. If you keep playing the odds, eventually they catch up to you. So I want to make a distinction here between 3 terms that you'll need to know what they are. The first is mutagen, M-U-T-A-G-E-N. That is anything which causes a change in the DNA. Anything which causes a change in DNA. Yes, ma'am.
Are you recording this?
Yes, ma'am. Number 2 is carcinogen. That is anything which causes cancer. Anything which causes cancer. By definition, pretty much anything that is a carcinogen is also going to be a mutagen. But they are distinct terms and they are typically used separately. And then the final one is teratogen. T-E-R-A-T-O-G-E-N, teratogen. A teratogen is something that causes a birth defect. So why do we use different terms for mutagen and carcinogen? Well, that is because of the different effects. Mutagen is the preferred term for a change in DNA in a germ cell. G-E-R-M. Germ cells are the reproductive cells. In other words, these are the changes that can be inherited. They can be passed on. Carcinogens generally refer to mutations in somatic cells. The somatic cells are the non-reproductive cells. So a cell in your eye would be a somatic cell. A cell in your stomach would be a somatic cell. A cell in the bone, you get the idea here. When you get down to it, there's actually only a very small number of cells where you can change the DNA and pass it on. Does everybody see that? A teratogen, by definition, does not, I repeat, does not change the DNA sequence. The gene is intact and it is perfect. Is everyone clear on that? But if you remember, I drew up the process of gene expression, right? DNA to RNA, to peptides to proteins. Everybody remember that? There's a lot of steps involved there. To go back to the analogy of building a birdhouse that I gave you then. If I had a master plan over in the Learning Commons, and that master plan is perfect, it has all the information on it that I want it to have, I send Case over there to make a photocopy of it. He comes in, trips, bumps in to Ethan, and Ethan accidentally spills ink on it. Has that changed the master copy? It's still intact over there, isn't it? But depending on how much ink covered that copy, you may or may not be able to build a full functional birdhouse from it. Does everyone see how that works? And the mutagen would change the master copy. A teratogen would affect something in the process between the master copy and the fully finished birdhouse. It's important to understand it does not change the DNA. If and when you ever become pregnant, they schedule a doctor's appointment, you go in and they talk to the OBGYN, right? And they basically give you a list of things you should and should not do. Don't drink, don't smoke, don't eat tuna, don't cuss. Why? Because it will negatively impact the development of the fetus. It does not change the DNA that's in there. It affects the development. Does everybody see this? Somebody here know who Billy Joel is? Remember that song he had, We Didn't Start the Fire, where he's just essentially rapping historical events that occurred in his lifetime? One of the lines in there is, children of thalidomide. Thalidomide is a medication, an antipsychotic, that was widely prescribed as a treatment for morning sickness in the late 50s, early 60s, especially in Europe. Within a very short time, they noticed that the rate of birth defects in women who were pregnant and taking this particular medication had skyrocketed. Essentially, rather than giving birth to healthy infants, they were giving birth to flipper babies.
I see. Actually.
Instead of having a full functional limb, they would just have essentially a flipper. It's important to understand those children had the correct genes to develop a fully functional limb. But since their mothers were taking this particular medication during their pregnancy, when the fetus was developing its limbs, the drug interfered and they didn't develop. Does everyone see how that goes? Remember when we talked about genetic switches? If the switch isn't thrown at the right time in the right place, that tissue will not develop. And I see one person writing that down. You have to have the switch get expressed at this proper time in the proper place. Y'all want a real life example of that? Okay. How many of you in this room actually began your journey as a human as a female embryo? I see a couple of guys looking around to see if they should raise their hand. Yes, you should. Because that is the default setting of every single human embryo. At a certain point, there is a gene called TDF on the Y chromosome that gets expressed. And it acts like a master switch and throws it off of this line and switches it over to this line. If that switch does not get thrown, it continues on developing as a female. If you throw the switch, but after you've passed the checkpoint, it will also continue to develop as a female. Pretty wild, isn't it? A good example of this is breast development. That path is started very, very early in the development of the fetus. Once TDF is expressed, that pathway gets shut down. This is why men have nipples but not breasts. Everybody see how it works? And that's just an example of a teratogen. The only one that I am aware of that they're really concerned about today, there's a drug called Accutane. Y'all may have heard of it. It used to be fairly widely prescribed for acne. I think it still is. These prescription guidelines used to require that if you were a female of reproductive age who received a prescription for Accutane, you also had to receive a prescription for birth control. That's still the case. It wasn't an option, it was mandatory. That's because Accutane is a very strong teratogen as well. I think they even used to go so far as requiring pregnancy testing. I appreciate y'all sharing because that's obviously not something that I would have knowledge of. Pretty wild, isn't it? Does everybody understand how this works and you understand the differences? I bring that up because there's sometimes confusion on what is a carcinogen, what is a mutagen, what does it mean? Well, now you know. And that would be a great list and describe question from the next exam. So, any questions over that? All right. How does cancer come about? Well, to understand this, you have to understand the cell cycle. The cell cycle is easy to draw. That's all it is. It's just a circle. It has 4 phases. We start in the upper left-hand corner on a clock, what would be about 10:30. Everybody see that? That first phase is called G1, which stands for the growth 1 phase, the first growth phase. During this, the cell takes in material and energy and grows. Small cell becomes a big cell. A lot of differentiated cells will never leave G1. If so, we say they are in G0. G0. That's why I drew the little arrow with the G subscript 0. Those are only for specialized cells. Is everyone aware of that, clear with that, and understanding of that? Stem cells will always move through the cycle. Specialized cells that have a specific function, you want them to stay in G0. You don't want them dividing. S stands for synthesis. We are going to double the amount of DNA that we have in the cell. This is when DNA replication occurs. DNA replication. It's the cell getting ready to divide. Why do you need to double the amount of DNA? Well, if you've only got one copy and you split, you're now left with 2 half copies, right? But if we double it and we split, each new cell has 1 copy. Everybody see that? After the S phase, it enters the G2 or second growth phase. Growth 2. And G2 is the final stage of preparations. We make sure we have enough protein made for the 2 new cells. We make sure we have enough organelles assembled for the 2 new cells. We double check that the DNA was copied and properly copied. We make sure we've got enough ATP on hand to not only start this process, but to finish it. It's kind of like the final checkpoint, the final countdown. Everybody see that? Then we move into M phase. M stands for mitosis. Mitosis. This is the division or reproduction of the nucleus. The division or reproduction of the nucleus. Is everyone with me? Notice I did not say it is division of the cell. There is a reason for that. There are some cells that have multiple nuclei, more than one. For example, muscle cells in a human, they typically have multiple nuclei. You don't want the cell dividing. You want to have those extra nuclei so you have extra copies to build proteins from. When you think of cell growth in the muscles, it makes sense, doesn't it? So everybody see that? In most cases, however, there is another process that occurs simultaneously called cytokinesis. 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 Cytokinesis is the division of the cytoplasm. The division of the cytoplasm. It usually occurs with mitosis, but it is not required. Do you have a question? And now, once all of this is done, we're right back to where we started. Except instead of 1 cell, we now have 2. I talked about Hayflick and his limit, didn't I? Yes or no?
Yes.
Okay. So the Hayflick limit is the number of times it can go through the cycle, essentially. And the reason for this is you start with 1 cell, you end up with 2. One of those can go on and be specialized. It can be a brand new cell, be pretty much anything you need it to be, right? But you're still left with a stem cell that can go through the cycle again as needed. That allows for growth and repair. If the cycle gets to the point where there's damage to a gene that controls the checkpoint, rather than staying in G0 where it should, the cell will instead begin to divide as rapidly as it can go through this process. We call that cancer. Cancer has 4 stages. That would make a great list and describe question, wouldn't it? Stage number 1 is accelerated cell division. Accelerated cell division. You start getting cells dividing when and where they shouldn't. Things start growing. They start getting bigger. At this point, the cancer is not a real problem. It shouldn't be growing, but it is. But it's not interfering with anything. And you remember we talked about the 2-hit model, right? So you've had to have 2 separate mutational events in the same cell. You knock out both copies. Depending on the tissue, depending on your genetic background, it may be really, really aggressive, or it may be really slow. It may essentially not move out of stage 1. You may end up never knowing you've got it. But if it's going to follow the typical path of cancer, it will then move to stage 2. Stage 2 is sometimes called tumorigenesis, or the formation of a tumor. At this point, there are generally going to be some signs that you've got something growing where it shouldn't. It will be visible, or it will be a mass or lump you can feel. It might be a mole on your skin that has started to get bigger. It may form a sore. Again, it's not really causing a problem, but it's kind of that there isn't something quite right. Why do I have this bump growing on my neck? Why is there a little bump under the muscle in my left forearm? I ask that because when I was about your age, I actually found a bump in my left forearm.
So it did not hurt? It just...
Didn't hurt at all. It was just a solid little mass. It was tiny. No flexibility to it. It just felt like I had something stuck under my skin, maybe the size of an aspirin or something like that. Just a small pill. Wasn't causing me any problems at all. Was not interfering with me. Didn't hurt. It just was a bump. Again, depending on the tissue and your genetics, it may grow, it may not. If it continues growing to the point where it invades adjacent tissue, we call this stage invasion. Stage 3 is it starts to invade the surrounding tissue. And generally at this point, it's causing some kind of issue.
So do you still have to...
I'm getting there.
Oh, no.
I'm getting there. It causes you pain. It restricts your mobility. Makes you feel weaker. You're not able to breathe as deeply or as easily as you used to be. You get an idea now. At this point, it is a problem. Then comes stage 4, metastasis. It metastasizes. Metastasis, Metastasis. Metastasis means the tumor begins to break off small chunks of it and get carried away to different parts of the body. It goes to a lymph node. It breaks off from your lung and lodges in your brain or your liver or in your bones. Does everybody know what happens at stage 5?
Yeah.
There is no stage 5 because at stage 4, it's pretty much a one-way ticket to death. Now, I want you to understand that this represents a continuum, okay? It's not you go through stage 1 and then stop. Then you're in stage 2 and then stop. Then you cross a gap and you're in stage 3 and then stop. It doesn't work that way. Like I said, it's a continuum. It just helps doctors classify the cancer so that they have a better understanding of where it's at and what your treatment options are. Can anybody tell me what the number one success factor in cancer treatment is?
Radiation
Nope, not the method.
or chemo.
Early detection.
Who said that? You are entirely correct. The sooner you find it, the easier it is to treat. And this brings us back to Case asking me if I still had that lump in my arm. So I will point to something and you tell me if you think I have it or not.
Looks like you don't have it then.
Looks like a scar, doesn't it? Because one afternoon I decided I got sick and tired of that. It's when I was over at school in Williamsburg. Jumped into my car, ran over to Pine Knot, and had one of the doctors there at the clinic take a look at it. And he said the only way to know exactly what that is to do a biopsy. But it's so small, I can take the whole thing out right now if you want to. I said, cool, can I watch? He said, sure. So I watched him cut this little thing about that big out, totally contained. Turns out it was a lipoma. Essentially, a tumorous growth of adipose or fatty tissue. It had just started to maybe enter into stage 2. I've not had any trouble with that arm since then. Yes, ma'am.
I have a question about tumors.
Ask.
Why do they grow teeth?
Why do they grow teeth?
Yeah, my mom is a social worker. She had a case where a kid had a tumor and it had teeth in it.
Because of the genes for the tissue that are involved. Why do we humans grow teeth? Because they're specialized structures that we use for a specific function, right? It would be nice when you reach my age to be able to turn them back on if you had a bad tooth, wouldn't it? But you wouldn't want to be growing teeth all your life, would you? You'd end up looking like Jaws or something like that.
I remember a person had like this kid or someone like had all these teeth as like upper jaw and such.
Because of what we said about genetic switches. You turn them on, you grow the structure, you turn it off. Some of those turn-offs are permanent, right? They never turn on again. Teeth are one of those. For whatever reason, that particular tumor, the particular genes that were damaged were in tissues that gave rise to teeth. They try to continue the specialization if they can. Does that help?
Yeah, thank you.
You're welcome. It's not a bad question. Feel free to ask anything like that anytime you want, because I want you all to be able to find some practical use for this in your own lives.
So our nails, so like they never turn off either? Like they just
No, because nails are a normal structure that has a normal function. Does anybody know what the function of a nail is? Protection. It protects your fingertips. But if you didn't trim your nails, and for whatever reason you were very careful not to break them, you can grow them out this long if you wanted to. Huh?
Those are disgusting.
Well, I don't think it's all that cool, but then again, I trim my nails.
Wouldn't that collect like a lot in germs?
Not necessarily, it's just dead material. It's just basically dead cells full of keratin, which is a protein. Hair, same thing, dead cells full of keratin. You ever exfoliated? You use the loofah? I was married, I knew what this was. What you're doing is you're sloughing off dead cells full of keratin. It's normal. Just the thing is that you have to remember about cancer, and it's not a bad question that you ask, Case. It's uncontrolled. There is no off switch. It typically is also going to be rapid. Remember I said it depends on the tissue, it depends on your genetics, where it's located, all that. This one just didn't happen to grow fast. I got fortunate. Some, they grow very fast. They're very aggressive. It's all in the genes that are affected. Any other questions? Yes, ma'am?
What's the most, is it skin cancer that's the most common form of cancer?
Oh, that's gross.
It used to be lung cancer. In fact, last time I checked, now this has been 20, 25 years ago, if I could have taken one action at that time to eliminate all forms of cancer, all forms, by a third, it would have been to eliminate tobacco use. That's why I do not understand anybody that smokes today, because it's basic death sentence.
It's probably just a dopamine effect to your brain. 'Cause
It's addictive, I know that. I would say it's probably going to be one of three things, and this is just me guessing. Now, I have a little bit more knowledge than you all, so it's somewhat of an informed guess, but it's still a guess. Probably going to be lung cancer because of smoking, air pollution, and the fact that it's incredibly difficult to detect. Lumps or bumps are not going to show in the lungs, are they? The second time, I would say, is colon cancer. For the same reason. Unless you have a colonoscopy, which literally is somebody anesthetizing you and sticking a camera up your butt and checking you out, you're probably not going to see it. And you eat a lot of carcinogens if you eat a lot of processed foods, and they can sit in your colon for days, can't they? And then I'd say probably the third would be skin cancer. Now, again, which one of those depends. People with darker skin are going to have much higher resistance to skin cancer than I would. Smoking rates have been going down here in the U.S. for 30 or 40 years. They've stayed about the same in Europe, and they keep increasing in Asia. A diet rich in red meat, that contributes a lot to colon cancer. And again, it's not a guarantee. Does that help? Yeah. Any other questions? These are all fine. They're great questions. Feel free to ask them. So early detection is your number one best thing. You find it early. You get a mole that feels like it's getting a little thicker, get it checked out. You're looking at a mole that starts to change color, get it checked out. It starts cracking and bleeding, check it out. You feel a bump where there shouldn't be one. And this goes pretty much anywhere in your body. You notice that you're starting to be very short of breath. You're coughing a lot. You can have blood when you cough. Get it checked out. So many people say, well, I know more than the doctor, I'm not going to go, and then they turn around and they've got late stage 3 cancer and they don't have a lot of options. It's unfortunate. And when you get older, you have to get some of these things done. Had a colonoscopy for the first time this past summer. Not the most pleasant experience I've had, not the worst I've had either. I got lucky. He said, no, you don't have any problems. You don't have to come back for 10 years. The prep was far worse than the actual procedure. So everybody understand the importance of detection? So what are your options for treatment? Let's list them. Number one treatment for any type of cancer, and I do mean, when I say any, I mean generic, because not all cancers are equal, right? But the preferred treatment, if they can do it, is going to be surgery. Because you cut it out, you throw it away, it's gone forever. Case in point, I've got a little scar, a good tale to tell my students, and no problems beside that. Surgery is not always possible, though, is it? My best friend's wife, she's 2 years younger than me, she developed colon cancer 2 years ago. They had to take out about 1 meter of her colon. That's 3 feet. They were fortunate because they were able to do surgery. Sometimes with something like that, or like lung cancer, it's very hard to have a surgical option. So you can't always use surgery, but if it's an option, you would prefer it because it's a guarantee. Second would probably be radiation. Radiation is where they essentially hit the tumor with a concentrated blast of high-intensity gamma rays with the idea that they're going to kill it and probably some of the tissue around it to ensure they got it all. It sounds far worse than it typically is. An example would be if you had a tumor located very deep in your brain. You don't want somebody cracking your skull open and carving your brain up like a fish fillet, especially if it's a small tumor, right? They are actually capable of focusing gamma rays so specifically, they could focus them in on multiple axes. They hit that one spot deep in your brain, area maybe no bigger than that, burn out all the tissue, kill it, and that's it. Now, it typically is going to have some side effects.
It's going to make you sick.
Probably make you sick. You might get skin burns on it. You are definitely increasing the risk of damage to the cells the radiation passes through, aren't you? But again, surgical options out. You don't want the brain tumor growing. It's late stage 2, but it's not to stage 3 yet. That might be your best option. In some cases, if you've got a very slow growing tumor, they will actually seed the tumor. I had a friend, he's much older than me, in mid-80s. He had prostate cancer 20 years ago. But his was a very, very, very slow growing variety. So what they actually did was they did microsurgery to implant tiny little radioactive pellets next to the tumor. And they essentially did enough radiation damage that pretty much the only thing it killed was the tumor. They pulled him out and he's fine today. It's pretty wild. Yes, ma'am.
Why does the radiation make your hair fall out?
I'm getting there. Y'all want me to talk about radiation poisoning as a whole? I'll do that after the third one then. Most times, if they do surgery or radiation, they will still probably give you a round of chemotherapy. Chemotherapy is just a polite way of saying poison. That's literally what they're injecting into you, is a low-grade poison. Cancer cells divide rapidly. The idea with chemo depends on if it's the old drugs or the new drugs. The older drugs targeted all fast-growing cells. Now, if you think about the main side effects of chemo, one of them is your hair falls out, right? The second one that most people would say is extreme nausea, followed by a complete loss of appetite, diarrhea, and you don't eat very much. That is because the epithelial cells in the hair follicles and lining the digestive system are the ones in the body that normally divide the quickest. At any given time, 90% of your hair follicles are actively growing. They typically grow for about 2 to 6 years before they enter the rest phase. And they're only in the rest phase for 3 or 4 months before they start right back up again. Y'all can tell I got my haircut, right? It was a lot better than that mop chop I had. And that was only about 6 weeks worth of growth that I had. My hair grows rather rapidly. And that is why whenever you get chemo or you get radiation, if you get a full body dose of radiation, those cells get so irradiated, it's so damaged, they can't stay alive. They die.
Do new ones get born? Do new ones get reproduced after they're gone?
Yeah, they will, but if you've taken too much radiation damage,
oh yeah, it's complicated, but
yeah. That's why typically when people stop chemo, their hair comes back, their appetite comes back, they're able to put on some weight. That's one of the reasons they tend to space it out and do it in rounds, because what you're essentially doing is hoping you can kill off all of the bad cells without too many of the good ones dying. That's why I say it's essentially low-grade poison. Those are very, very potent drugs. And that is generally what cancer is and generally how it's treated. Can you cure cancer? Not really. What they say is remission. You're considered to be in remission if after 5 years you haven't had a recurrence. In other words, you've not had a new tumor pop up somewhere. I think for some types of cancers it may be 10 years, but you get the idea. What that essentially is saying is you are no longer considered at a higher risk for cancer than a normal person, your normal lifestyle, your age. Until 5 years have passed, you are considered at a higher level risk. Is that clear?
so just the cure is just to either remove it or just kill it.
Yeah. I knew someone that had early stage 3 thyroid cancer in 2013. Removed the thyroid, did radiation treatment, did 6 months checkups for 5 years, goes back once a year, still to this day. Here we are, 13 years later? And the last appointment I went with him, the doc said, your baseline, there's no need for you to come back every year. You can if you want, but you're not going to get a recurrence of it because we got it all. Otherwise, you'd have seen it by now. Cured? Depends on how you define cure, doesn't it? I like what you said best. Basically, they got rid of it, killed it all, so it won't come back. So, any questions about cancer? Last time I checked, which again has been some time, cancer was the number 2 killer in America.
Can I ask what's one?
You can ask. I'll see if you can guess. It should be kind of obvious.
Car
Well,
crashes?
huh?
Car crashes?
Nope.
Old age?
Old age.
Oh, just no.
Old age is actually cause of death. It's generally organ failure due to old age. Now, heart disease is number one. Heart attacks, high blood pressure that causes kidney failure, strokes, all of that is under the heading of heart disease. That's been number one for a very, very long time. It used to be infectious diseases were probably half of the top 10, but by the mid 20th century with vaccines and antibiotics, they pretty much wiped that out as even being in the top 20. And heart disease, there are so many things that go into that. It's, again, difficult.
I know if you're like over 300 pounds, that your heart gets more affected.
Sure.
Yeah.
Who has a bigger risk of heart disease? Me or Ethan? Why? I'm older. You look like you're probably bigger than me, and stouter. And I don't mean anything bad by that.
No, I see where you're going with this though.
The point is, it's really difficult to say it because you don't know my family history. I'd say you're right because I have a family history of hypertension. I am under treatment for hypertension. That's high blood pressure. I also have tachycardia, which is a rapid heartbeat. Normal is somewhere between, say, 65 and 80, in that range, sitting still. If I'm sitting still, completely relaxed, meditating and doing breathing exercises, I might get mine down to 90.
My mom has like this condition, like she recently got it like last year. It's called POTS. You ever heard that? She recently got it. Like she never had like any issues until like after a Florida trip and it started hitting her heart.
This is getting back to what you were asking about old age. I am definitely older than you, so I am probably going to have more conditions. I don't normally share health information with just everybody, but you all seem interested, right? You know, I have type 2 diabetes, right?
Mm-hmm.
So black coffee, no sugar. When I got myself a soft drink out of the machine over in Cooper Hall for drinking with my lunch, do you think I got my preferred Dr. Pepper? Sugar-free. I brought it with me. Zero sugar, sugar-free diet, whatever. I have to take something called metformin twice a day. Once in the morning, once at night. I also track my steps. I don't know if you saw me tapping my watch a couple of times just to make sure I'm getting enough exercise in. Fortunately, I don't have to give myself insulin injections very often. In fact, I think the last time I had one, I got one last week because I was showing my anatomy and physiology class what happens when a normal person eats a candy bar and what happens when I eat a candy bar and drink a 20-ounce Dr. Pepper. Yeah, I jacked my sugar way up. Took one for the team. They had to see it, didn't they? And then I gave myself an insulin injection so they could see what that looked like. And it shocked them how much it dropped it in just an hour. I dropped it almost 100 units. But as I told them, that's because I generally have it well managed. I knew exactly what I was doing and how much I was taking in. I also knew how much insulin to give myself. I have high blood pressure. I have to take 3 different medications for that, and I have a 4th that I can take if I find it going up. And I know the symptoms of it. I start to get hot, I start to feel flushed, I start to get red, and I start to get a headache. It's probably elevated right now. Y'all causing it to go up. But if I hadn't shared that, you wouldn't have known it, would you? And believe it or not, despite those conditions, I'm considered to be in generally excellent health for a man my age. Comparing me to you, I guarantee you, you probably don't have to take anywhere near the number of daily things that I do. And that's fine. I'd hope you wouldn't. Have to, at age 20, hear something wrong. You have a question? Okay. It's okay. I just keep seeing the hand go up and I keep hoping, do you have something to say? Main thing is, if you find it early, you've got a lot of options. They are really, really getting more and more sophisticated with their treatments for cancer. Probably the biggest one in my lifetime that I would say is the coolest would be the treatments they had for leukemia. When I was a child, leukemia was a death sentence. If you got it as a child, you wouldn't see your 10th birthday. In the 80s, they developed bone marrow transplants, and now it has one of the highest rates of remission of any kind of cancer, if you can have a donor. What they do is they blast the bone marrow with high intensity radiation to essentially kill everything. It kills all of the cancer, but it also kills all of the stem cells in your bone marrow that make white blood cells and red blood cells. But if you've got a close relative who's going to be a match for you, they stick a big needle into their bones and they pull out the bone marrow and inject it into you. If it's successful, you essentially reboot your immune system with their cells. How cool is that?
Isn't that like super painful?
Yeah, but if my brother came to me and said, hey, I need a bone marrow donation, I'd probably say, yeah, because I might need one someday too.
Does it make your bones like weaker for a while?
No.
They're just,
Believe it or not, there's a process called bone remodeling that goes on. And I can't remember if it's every 3 years or every 7 years, you essentially replace every cell in all of your bones with a new one.
How do they like test to find out if like you're a match?
They generally will do a blood draw and they will see if, basically, if you put my blood with your blood, is there an immune reaction? It's a little bit more detailed than that, but that's generally a rough idea how it works.
so
That,
it's
well,
basically,
hands up if you have a question.
So would that mean that they would have to have the same blood type in order to do it? Or?
Probably so.
Okay.
so it's just like a blood transfusion to somebody else, boom, basically. Right?
Pretty much. See, I could give you my blood. I've talked about that, haven't I? I'm the universal donor. But they would still screen my blood if they were going to be doing this as a general rule. Let's just pretend yours is type A. We don't know, but that's what we'll say. Is that fair? If there's an emergency and they don't know what yours is, they know they can take it from me and give it to you and save your life. However, once they type you and they figure out your type A, Zachary over here, who's type A, might be a better donor for you. And I hope you don't mind me using you. Everybody see how that goes? So questions? Okay. Let's close the book on cancer then. I think that's a good little lecture. One last thing about cancer cells, they're effectively immortal. I talked about that already, didn't I?
I think
Telomeres, telomerase. That's what makes them so deadly. They divide so rapidly and they are effectively beyond the Hayflick limit.
so. If you got rid of them or like killed them, can they like are they still basically alive, immortal, anybody?
Yeah, if they took them out of me and I died tomorrow and they keep it going in cell culture, there could be cells of mine living 50, 60, 70 years from now. Y'all don't believe me? Did I ever tell you the story of Henrietta Lacks? HeLa cells? She died in 1951 from cervical cancer, and her cells are still alive in labs all across the world today, 75 years later.
You did tell the story, yeah.
Yes, sir.
What would like the cell culture be? Would it be just like glucose and stuff?
Yeah, they basically, you have a little bottle that you lay on its side. It's just plastic. And you fill it about half full of what we call growth medium. And the growth medium is just a liquid that contains everything the cell needs. So it's going to have some minerals in it. It's going to have some glucose in it. It's going to have some ions in it, a little bit of salt. And the cells will actually lay down an extracellular matrix and start sticking to the bottom and grow as kind of a film on the bottom of the flask. And whenever you're ready to subculture it, let's say you do it every 2 weeks. You put a little bit of trypsin in there, which is an enzyme that breaks up that ECM. You tap it gently, pipette it up and down, and the cells start to float. And you just take a few milliliters of the liquid and you inject it to a new bottle and you put it right back in the incubator. It sounds cool, but when you do it a couple times, you're like, this is really just kind of like no different than scrubbing your toilet every other week. You do even the most wondrous things and they become routine, I assure you. I remember the first time I did DNA sequencing, I could not wait to see the results. The last time it's like, man, I wish I had $1 every time I did this.
So it's more fun than actually doing it, like explaining it.
Well, the point is, familiarity breeds contempt. I'm sure you've heard that, haven't you? Anything can become routine. So that's it for cancer. Now let's talk about mitosis. Mitosis itself has 5 phases. I like to joke that I can always tell if a fellow biologist is a geneticist or a cell biologist by how they describe the cell cycle. If they describe it like this, they're a cell biologist because they're interested in the whole thing. A geneticist will say you have the 4 stages of mitosis and the rest is all interphase because they're interested in the DNA. Interphase literally means between phases. It is the G1, S, and G2 all lumped together. Interphase is G1, M, and G2 all lumped together. It's just a way of acknowledging that there are other things going on here, but it's not necessarily viewed as that important by that particular researcher. It's arbitrary. It's kind of funny if it weren't so sad. I do both and I laugh at both sides. Yes, we scientists have humor too. We tell some strange jokes. Don't worry, I won't pull one on you. So everybody with me? The 4 phases that are of interest are what really happens in mitosis. So let's assume that we have just started mitosis. We've just moved between the G2 and the M. So we are right here. Everybody with me? The first thing that's going to happen is called prophase. Prophase. During prophase, 2 things are going to happen. Number 1, the chromosomes will condense. The chromosomes will condense. That means that they coil up around proteins and they become visible. Normally, when you look at a cell, you might see the nucleus, but you will not see the chromosomes aligned in there. Is everybody clear on that? That's just because they're open. The DNA is uncoiled, it is open. You're allowing RNA polymerase to come in there and make RNA copies. You want to be what we call transcriptionally active. You want your genes to be able to be expressed. When they're in prophase, they're all closed up, supercoiled, and you cannot see the expression. The second thing that occurs is the nuclear membrane will begin to break down. The nuclear membrane will begin to dissolve. A structure called the spindle is going to form. The spindle just kind of looks like a network of threads. You've got two little poles here on either end, and threads start to come out from it, and the chromosomes attach to it. The chromosomes attach to it. Am I going too fast? There's a lot going on during prophase, but it's essentially the start of the process. You got to get the chromosomes coiled up and ready to go. The nuclear membrane is in the way. You want to break it down so you can have this spindle forming, and the chromosomes are going to attach to them. Is everybody with me now? That brings us to phase number 2, which is called metaphase. Metaphase. M-E-T-A. Isn't that the name of Facebook now?
I thought that was for the AI. Yep.
AI runs the world.
Yes, it does.
Metaphase is actually the easiest one to see because all of the chromosomes are now attached to the spindle and lined up in the very middle of the cell. And I mean, it's very obvious. They're all right there in the middle. And what's going to happen next is something, what did you say?
No, nothing.
Called anaphase. A-N-A-P-H-A-S-E. Anaphase.
So that's phase 3.
Phase 3. In anaphase, the chromosomes are actually going to look like little arrows pointing away from the center and towards the ends of the spindle. They're actually going to look like little arrows like this, just kind of drawing away. And they're getting pulled apart. The chromosomes are being pulled apart. And the reason for that is because they are being pulled towards opposite ends of the cell. They are being pulled towards the opposite sides or ends of the cell. Why do we do that? Well, because we doubled the amount of DNA, didn't we? We're wanting to now separate it. We're wanting to ensure that we have the same amount over here as we have over here. Everybody see that? And that brings us to stage 4, which is called telophase or telophase. T-E-L-O, telophase. Telophase is essentially the reverse of prophase. So you're going to see the chromosomes will decondense. They're going to relax, they're going to uncoil, they're going to go back into their non-visible state. that spindle, which kind of acted like a motor, just drawing them in, it's now going to break down because it's no longer needed. And finally, the nuclear membrane is going to reform. But instead of having one big super large membrane like this, you're going to have two separate ones on either end of the cell. And how many nuclei do you have now? Two. I want to emphasize something here. There is another process that generally takes place at the same time as telophase. However, they are separate processes and they don't have to occur at the same time. Is everyone clear on that? Thumbs up? Okay, this process is called cytokinesis. 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 It literally means division of the cytoplasm. You draw a line right down the center and say, you keep everything on that side, I'll keep everything on this side, and we are now two separate and distinct cells that are called daughter cells. They're called daughter cells. They are genetically identical to each other. Now, why would cytokinesis not occur? Because, as I said earlier, there are some cells that have multiple nuclei because it allows more efficient gene expression. It allows more efficient gene expression. If I sent every single one of you over to the Learning Commons to check out the master plan of the birdhouse, photocopy it, check it back in, put it away, and march back over here before the next one could go over, it would take you a while, wouldn't it? If I set a stack of them right there on the front desk, and there's 20 in the stack, and I said, grab one and go wherever you want, that's going to be a lot quicker for everybody, isn't it? So certain cells have multiple nuclei because it makes gene expression more efficient. Best example I can give you would be human muscle cells. Human muscle cells. And you think of how easy it is to pack on muscle when you're working it, it makes sense, doesn't it? You need a lot of protein. It's one of the reasons they say eating meat is such a good thing for getting protein, because it's very rich in it. Muscle cells even have their own special form of globin to help make them more efficient at using oxygen. Did you know that? You ever had a steak and you get that runny red juice out of it when it's fairly rare? That's not blood. That is a protein called myoglobin. It's what gives muscle its reddish color, or red meat its reddish color. Pretty wild, isn't it? And how about we stop right there and I hand back your exams.